And also we had approval by Brazil's Anvisa in April. So along with our partner Janssen Pharmaceuticals, we continue to work with additional health authorities around the world to bring this treatment to patients around the world. And we're very pleased to share the data from the ongoing CARTITUDE program, including longer follow-up of our CARTITUDE-1 study, which was the basis of the US FDA approval and also EC approval. And also data from Cohort A and Cohort B of the multi-cohort phase 2 CARTITUDE-2 study. So these heavily pretreated patients with multiple myeloma face poor prognosis. The recent approval of CARVYKTI, together with the updated data from the CARTITUDE program, continue to expand the treatment landscape for patients who otherwise would have very few options. So thanks again for joining us, and it's now my pleasure to introduce Dr. Sunda Jagannath, Professor of Medicine, Hematology and Medical Oncology at Mount Sinai School of Medicine and Director of the Multiple Myeloma Program at Mount Sinai Hospital. With that, I'm going to turn over to Dr. Jagannath, please.
Thank you very much for the kind introduction. And I thought you said it was forward-looking statement, but it looks like I'll have to do a forward-looking to look at the full slide. I'm not tall enough, I guess. All right. So today, in the ASCO yesterday, we had shared the information of CARTITUDE-1. This was the 2-year post last patient in. The median follow-up is 28 months, but the last patient in had a minimum follow-up of 2 years, 24 months. So this is an update. And this has a 6 months extra follow-up compared to the data that was reported in ASH 2021. So as an introduction, you're all familiar with the CAR construct for this particular product, cilta-cel. It has 2 binding domains, but this is a variable heavy chain gene, and it binds to BCMA at 2 different sites. So we believe that either that 2 binding, it helps bind it tightly, or it is able to find even lower expression of BCMA and the myeloma cells. And that may be part of the reason why it is much more efficacious, even in patients with low level of BCMA expression on their tumor cells. So in the ASH, with a median follow-up of 21.7 months, we gave you the overall response rate and stringent complete response rate. Even with further follow-up, you can't change the overall response rate because overall response happens very quickly with this drug. So there is no change in the overall response or the stringent complete response rate. But you will see that the median progression-free survival is still not reached. That's the most important information for today, because I think there was a presentation in ASCO where they had used an earlier time point with 1 year follow-up where it was projected the median follow-up, the median progression-free survival would have been around 2 years or something like that. But now with a more mature follow-up, we are actually here to show you that the median progression-free survival is not reached, and therefore the overall survival is not reached. And this is very important. You will see the study was actually designed with patients who have had 3 or more prior lines, but the median number of prior lines was 6. So the FDA looked at it and said, okay, then we better have 4 or more prior lines of therapy. So this is true for both the cilta-cel as well as for ide-cel approval. It is 4 or more lines of prior therapy. But the patient should have been exposed to the most important classes of drugs, which is proteasome inhibitor, immunomodulatory molecule, as well as anti-CD38 monoclonal antibody. And while this was approved this year in February in United States, I think they also have, European Commission has granted them conditional marketing authorization for CARVYKTI for the treatment of adults with relapsed and refractory myeloma who have had at least 3 prior therapies. So it is slightly different in Europe. And of course the same including PI, IMiD, and anti-CD38. With that, I will move on to show you what it was. The study design, you're all very familiar. There was a phase 1B, which was basically to show the safety of cilta-cel and confirm that the doses which was used in China, then it was brought over from the United States, that was the appropriate dose. And the dose is actually shown there. The target was 0.75 million CAR T cells per kilogram body weight, but actually on the clinical trial, 0.71 million cells, viable CAR T cells were administered for these patients. And the phase 2 is what resulted in the approval of the study, and the key eligibility criteria are shown here. All the patients did receive cyclophosphamide and fludarabine before they received it. That is the lymphodepletion, so that the CAR T cell doesn't get rejected. So they all get the lymphodepletion. Then they get the cilta-cel infusion. And this is one and done approach. It's almost like an auto transplant. You give CAR T cell. There was no further treatment after the administration of the cilta-cel for the patient. So what do we have here? The efficacy is, again, overall response rate is 98%. So the total number of patients treated was 97. So 95 of the 97 patients responded. The median follow-up is given there, 27.7 months. And I told the minimum follow-up, the last patient in was followed for 24 months. And you could see that most of the patients who were evaluable for MRD, 92% were MRD negative at 10 to the minus 5. And since the response rate was so high, 98% overall response, stringent complete response rate was 83. So this is also indeed remarkable. Once you have this kind of response rate, then basically the response happens in all patients. You will see that when I show the subgroup analysis going forward. So here's the efficacy outcomes in the subgroup analysis. The top one, it is a busy slide. I will just show you. First is the overall. 97 patients were enrolled, so that's what it shows there on the top line. And the overall response rate, as you saw, was 98%. And you see in that one, 61 patients were evaluable for MRD negativity, 56% were MRD negative to 10 to the minus 5. Next-gen sequencing, 91.8% MRD negative. And you can see 27-month progression-free survival. You could see it is, 55%, so we still haven't reached. And I will talk to you later on when I show the graph what I perceive will be there. And then at 27 months, you see the overall survival is, 70% there. So 27%, 70% of the people are alive. That's important. Now then, the next one is, did the age make a difference? What about people over the age of 65? You could see almost 35 patients were there who were treated, so it's a sizable number of patients, and you could see that their overall outcome, 27-month PFS and 27-month overall survival was excellent in this group. Since everybody responded, I didn't talk about the response. African American, 17 patients or 18%. The United States, the African American population is 17%, and that's what the white paper FDA had put out, that they expect that all the clinical trial coming in front of them should reflect the U.S. population. So this one does reflect the U.S. population, and you can see that in this subgroup of patients also, the overall response rate is 100%, and the 27-month PFS, their median is also not reached because it is 51.8%, and the 27% overall survival is also very good. And patients who had 3 or more prior lines of therapy, 4 more, none of that made a difference. Triple class refractory, penta-drug refractory. So what I wanted to show is penta-drug refractory. You have 41 patients, penta-drug, almost, closer to half of the patient are penta-refractory, and yet you could see that the overall response rate was 95%. And their 27-month PFS and 27 overall survival. So the 27-month PFS is 61.6, so their median is still not reached. So even in the penta-refractory, this was really a life-saving treatment for these patients. Then let's go through cytogenetic risk. High-risk patients, you could see the response rate is 100%. The median PFS here, it is, shorter. It is 21 you can see that 21 months median PFS. And so 27 months, only 43% of the patients are progression-free, but the overall survival is still very good. 65% of them are still alive, saying that some of these patients have been rescued subsequently. The ISS stage 3, it's a very low number of patients, 14 patients. So I would not get swayed very much by this particular data, because ISS stage 3, at the time they were entered into the study, basically reflects high tumor burden. And we will talk about high tumor burden per se, and you could see that 27-month PFS is 34 and 50%, so their median PFS is also shorter, 15 months. Then the extent of bone marrow plasma cell infiltration, those who had packed bone marrow greater than 60% plasma cell infiltration, there were 21 patients. Those patients also had a shorter median progression-free survival. As you can see, median progression-free survival was 24 months. The baseline BCMA expression did not matter, and that, as I told you, was because of the receptor. They were able to, there were 2 receptors for BCMA on the CAR construct, so it was very effective even for low expression of BCMA. And presence of baseline plasmacytomas, extramedullary plasmacytomas, you could see 20 patients. Overall response rate is 100%. This is very important because they will say extramedullary plasmacytoma, bad outcome, whether Silter cell is better or ida cell better. You got 100% response. All these plasmacytomas disappeared. And that's very important. That means the treatment was effective, But these patients had high-risk disease. Later on, they relapsed elsewhere. That is not unexpected, like any other. But the fact is, was Silter cell effective? It was effective 100%. All the plasmacytomas disappear. And that is very important for us to remember. That's the key message there. Now, going into the continued progression-free survival, this is the important update. We'll go from the bottom red line on the PFS. That is the PFS for all patients. The PFS is not reached. As you can see, 54.9% of the patients are still progression-free and alive at 27.7 months. So if you look at that, the longest patient, this patient will be 4 years by August and will come to my center for a follow-up visit. This patient is still doing very well, and I'm very sure this patient will be in complete remission, MRD negative, because the patient is serologically negative. The next 2 patients are also mine, 3-year follow-up. One of them went through next-gen sequencing. She's completely MRD negative, fully active, from New Mexico. And then there is another one, which is actually a physician from Florida, and she's also doing well. She did not come because of the COVID spike in New York, but she'll be coming in July, and she'll go through MRD testing. It would be remarkable that if she's also MRD negative. Why I wanted to point this out is, once you are MRD negative every year for 3 years and 4 years, then you are reaching a kind of a different level of dropout. So the PFS is not going to be a steady decline you will project. That's what they tried to do earlier, projecting, the PFS for, Silter cell will be 21.4 months, things like that. But we saw, as we followed, it is not. And this is why, even in the 6 months follow-up, we thought it would have dropped, but it didn't. Because these patients were consistently MRD negative, and didn't go through any of the complications. They are doing very well and good quality of life. The overall survival is excellent, as you can see, and even in the PFS, you can see patients who are MRD negative at 6 months or MRD negative at 12 months, you can see it goes higher and higher. So if you look at the PFS rate of 78.8%, close to 80% of the patients are PFS free if they were MRD negative at 1 year. That is remarkable. Those patients are all now 2-year and 3-year MRD negative. And the overall survival shows that if they achieve MRD negative and they have sustained MRD negativity, then they are looking at long term. So I would only use the term cure. If these people stay for 5 years MRD negative, then I usually will call them, at that time they are cured, if they are MRD negative consistently. Now we are doing the MRD negative at 3-year mark. So safety. The safety profile was manageable with no new treatment-related death. What is important is a total of 20 second primary malignancies were reported in 16 patients. So 16 patients at second primary malignancy. And this included, none of them were related to Silter cell, but let us look at it, because obviously 20 second primary malignancy in 97 patients, that's almost like 20%. Isn't that very high? Usually we are used to looking at 6% with Revlimid maintenance post-transplant. 20%, and you say it is not related to Silter cell. Is it your investigators just don't want to blame the drug? So that's an important question. So if you break them out, you could see that 6 of them are non-melanoma skin cancers. In this patient population who have been heavily pretreated, especially if they had prior lenalidomide exposure, etc., they can get squamous cell cancer, basal cell cancer. So this is not uncommon in these patients. Then you also saw that there were 1 patient each with malignant melanoma, adenocarcinoma, myxofibrosarcoma, that's a uterine cancer, and a prostate cancer. So in this patient population, having a prostate cancer or a melanoma, it is not uncommon. And I don't think this was related to cilta-cel, but you can always say, does the immunosuppression still has a role? But that is something we have to see. Then there are 9 patients with hematologic malignancy, 1 low-grade B-cell lymphoma, 6 MDS, and 3 fetal AML. So that kind of intrigued. The question was asked, 3 fetal AML, isn't that pretty high in this patient population? So this is important because the median follow-up, when we looked at it, the 3 fetal AML, we had reported on that at 1 year follow-up. Now we have 1 year follow-up on CARTITUDE-2, and I will go through that. We have not, 17-month follow-up and 1 year follow-up on CARTITUDE-2A and 2B. We have not seen that AML. Their hematologic recovery is very good. So we have to see whether this was related to this heavily pretreated patient or not, and that signal is ever seen in an earlier patient. So far we have not seen the signal that quickly, because these all happen in median 1 year follow-up. We were already seeing this. So that's important to see whether it is the case or not. Then, 1 new case and signs and symptoms of Parkinsonism on day 914. So 914 will be about 2.5 years, because 365 days, 730, and then it'll be 1030. So it'll be about 2.5 years, somewhere thereabout. And so there was 1 case of Parkinsonism. So going back, we had reported 5 cases of Parkinsonism initially to you, or Parkinsonian-like, or there was another MNT and terminology, but FDA made it easier for all of us. So because the physician can recognize Parkinsonism rather than MNT. So Parkinsonism-like thing. So of those 5 patients, we had reported 2 of them died of infection. 1 patient actually died of Parkinson neurological complication. Two patients were there. One of them, the symptoms completely disappeared. 1 is improved. And this particular patient, actually, I talked to the physician without saying the physician name or location. That patient apparently is going to Grand Canyon to hike. So yes, he has tremor, but physiologically he's doing well, and he's in complete remission, MRD negative. But it is still of interest to us to see. And this particular patient, he is the information I can tell you is that he had 2 of the 4 symptoms ahead that gives rise to the Parkinsonism. And we had a mitigation strategy for that, and we will talk a little bit more about that. But in conclusion, I can tell you at 28-month follow-up, patients were treated with cilta-cel, maintained deep and durable responses with median PFS and OS not reached, and manageable safety. And so I wouldn't start projecting on the median PFS, because I told you the longest one, the 4-year follow-up, is still in complete remission. The next 2 patients on that dotted line, they are mine, and they are, one of them confirmed MRD negative at 3 years. The other one will be coming up in a month in July. So they are all doing very well. So the progression-free survival curve is changing. So that is important to keep in mind. The safety profile is manageable, and a favorable risk-benefit profile is there. So let's talk about, there are currently further investigation in myeloma in earlier lines, CARTITUDE-2, 4, 5, Imagine, CARTITUDE-6. I'll just give you some update on CARTITUDE-2. By the way, the data which was presented here is already out in JCO for those who want to look up the manuscript for additional details. So CARTITUDE-2 study primary endpoint is minimal residual disease negativity. We were interested how many patients will go into MRD negative. So these are 2 cohorts, cohort A and cohort B. The goal was to accrue 20 patients on each, but 20 in cohort A and 19 on cohort B. So cohort A, these patients have had 1 to 3 prior lines of therapy earlier, and they should be lenalidomide refractory, because investigators from Mayo said lenalidomide refractory meant that they had a poorer outcome, but these are all single institution trial. Whatever it is out there, so we will take it with a pinch of salt. So lenalidomide refractory. And then comes the cohort B, 19 patient early relapse. These are patients who are in frontline therapy right after the transplant. If they relapse within 6 months, etc., they are all considered high-risk patients. So these are early relapse, 1 or between 1 and 2 lines, you have to say, because they relapse after the first line of therapy very quickly. So the schema is exactly the same, and the secondary endpoints, of course, we are interested in overall response rate by IMWG criteria, and duration of MRD negativity, etc. So what did we find? So cartridge cohort A, this is the 1, 2, 3 prior lines. Again, we were able to see overall response rate is 95%. But more important in this patient, because the aim was MRD negativity, stringent complete response, yay, we got 90%. That's very important for us because that's one of the objective of the study. So the treatment is quite effective. You can see that. And, 15-month PFS rate was 70%. Okay. And so that means, has there been relapse? Of course, there have been 2 progressed and died. As you can see there were 4 deaths occurred, 2 due to progression of disease at 426 and 550 days. So both of them are more than 1 year out after the CAR T cell therapy, and 1 due to sepsis and 1 death due to COVID. This COVID era is a problem when we are conducting clinical trials. Anyway, coming back to that, the safety was manageable, including 1 patient treated in an outpatient setting. CRS, again, most patients, I tell them they will have a fever. Actually, the patients pray for fever because once they have fever, then everybody is smiling. Okay, it is going to work against my cancer and I'll be doing good. So CRS is kind of universal with this particular cell-to-cell product, because you will hear more about other products where they are not even having CRS. So yes, there are other products with less CRS, but here the CRS also is a good indication for patient the treatment is working, because it is a high CRS rate, but it is all grade 1 or 2. It's mainly fever. You can see grade 3 or more is only 10%. Neurotoxicity, all grades is 30%, but grade 3 or 4 is only 5%. And there was no case of Parkinsonism in this earlier cohort of patient. And cohort B, you could see there were 19 patients here. Overall response rate, 100%. That was again the goal, because MRD negativity was the major goal of the particular study. That is the primary goal. And then you could see complete remission or stringent complete remission is greater than 90%. Fantastic. So that gives us a lot more confidence it'll reach the goal. And as you can see here, it is all very fast response rate. 12-month PFS rate was 90%, because here, the follow-up is only 13 months, median follow-up, so a lot more follow-up. You will continue to hear in EHA and ASH and things like that. So you will get every 6 months, you'll get an update between ASCO and ASH. And of the 15 patients with MRD valuable, you could see 93% were MRD negative, which was one of the goals of the study. And the safety was manageable, including 1 patient treated in an outpatient setting. CRS, again 84%, grade 3-4, again low, 5%. Neurotoxicity, 26%, grade 3-4, 5%. But 1 patient did have Parkinsonism-like symptom. But here we did have a group meeting, safety, because all the investigators are called whenever something happened. This was a young patient in his 40s, treated in, I can't tell the country or place, but, outside of United States. This patient was progressing through had the CAR T cell therapy acquired, but then the patient actually was progressing through what we call as the bridging chemotherapy. And he was in his 40s, so the doctors decided to give. So exactly what we said, mitigation strategy. What was the mitigation strategy? How could you predict in this, and what went wrong in this patient? So one patient presenting with high tumor burden, and patient presenting with grade 2 or more CRS, patient presenting with any stage ICANS, and high expansion of the CAR T cell and persistence of CAR T cells. What was the mitigation strategy? So in order to avoid that, we said that you wanted to have the disease under control, so use effective bridging chemotherapy, bring the disease down, then monitor the patient closely by handwriting, etc., and treat the CRS and neurotoxicity effectively. In this patient, because he was young and the tumor burden could not be reduced, it was one of those preventable. So I would have to say, at least what is good is we were able to identify the 4 reasons, 2 of the 4 factors which can give rise to Parkinson type thing. And if you follow the mitigation strategy, you can mitigate it, because the incidence was higher on cell-to-cell 1, in the cartridge 1 trial, but it is now down to less than 0.5%. So it has really gone down, but we did come across it, and it again tells us we have to be vigilant. So in summary, overall response rate 98%, in the CARTITUDE-1, and you can see 95%, 100%. But for CARTITUDE-2, the most important for us was stringent complete response, because the objective of those trials was MRD negativity. So having 90%, stringent complete remission was very, very important. I would stop here.
Q&A.
Thank you, Doctor. This is Gina Wen from Barclays. So I have maybe 3 questions. The first one is regarding the Parkinsonian. So just wondering why, one patient will onset will be after 2.5 years, and then I think a CAR T cell should already, majority of them maybe already disappear. So why that will happen, late onset? So that was the first question. So my second question is, you comment on the all grade and a grade 3 and 4, the CRS, and I'm just wondering what is the rate of a grade 1, and also what is the rate of a CRS that occur after 72 hours? And my last question is for company. I think our channel check suggesting...
I think we'll give you time to ask the next question. Okay. You keep the mic so you don't have to worry that it's going to be taken away. Okay, we will answer all 3 questions. So let's start with the first question first. So your question was why delayed Parkinsonism disease. This is very important because this was for us, at least for me, it was the first in human in the United States. It was done in China, but we were making sure there was not, and it is our first experience, and we were following the patient long enough to say. Initially, we did identify Parkinson type reaction happen, and then we put in the medication strategy, and we thought that at the end of 1 year, we had not seen any noise. We were all very happy it was not happening. And then now we realized this particular patient did develop a tremor, and he had a spinal tap. There was no CAR T cell in his CSF. CSF was clear. He, in the peripheral blood, he did not have. So we do know that the cilta-cel by vector copy in the peripheral blood does disappear very quickly within 6 months. Probably even as early as 2 months, but by 6 months it is gone. So that's why it is almost like a one and done deal. It is not a matter of these cells staying there. Now, in science, how the scientists always hem and haw. Well, we do not know whether in the microenvironment some of these cells are still there in the bone marrow somewhere, because all the BCMA expressing plasma cells disappeared, and these cells, few are there, and then if the BCMA goes up, they come up. But we have not seen that at all, that any kind of increase in the cilta-cell or vector copies, and in this particular patient there is not. So then the hypothesis has to be, this patient did have the 2 of the 4 factors, high tumor burden, and he also had grade 2 CRS, and so he had 2 of the 4, ICANS and CRS, so 2 of the 4. So maybe he had something subtle then, and then later on it manifested, and now it is going to go away because he's hiking and he's doing well. And we know among the other 5 patients, 2 who were, one of them is actually resolved. So it looks like the neuron is also able to regenerate or cope with it. So it looks like if it was, the damage was not very severe and early in the process, then the patient may recover. But this is an observation we are learning. Now the next step is for us to see any other case happens and go and look back. But beyond that, that's as a physician taking care of patient, that's all I can tell you right now. Your question number 2 was the CRS grade 1 and 2. As I said, the 95% of the patient had CRS, and the grade 3 for CRS was in the neighborhood of 5%. So yes, everybody gets a fever, and you got to remember the fever is indication because of the new thing as considered as CRS grade 1 in the new classification. They simplified it, so fever becomes a CRS. So everybody gets a fever, and in this particular product, both the doctor and the patient like to have fever.
If I can add, because our CRS starts like week after infusion, so because your question was also in first 7 hours, so fever is start of CRS. So in average, it starts 1 week after
day 7,
day
the
7.
time when it comes. And that is because of the central memory cells. This is much more expansile and more deeper, and it takes time to expand and gives that. But they are monitored How it happens, if you actually monitor the thing, first is T cells expand, and when they expand, they have to produce IL-2. So the cytokines, IL-2, gamma interferon, GM-CSF, TNF-alpha. So the cytokines come first. Because the cytokines, then the body reacts to it. IL-6, immediately CRP will go up, gamma interferon, then immediately ferritin goes up. So clinically, we physicians follow the CRP and ferritin to follow. So that follows the cytokine. And then cell expansion actually even happens further down, day 10 to 14. So this is almost very predictable in all CAR T cells, because that's one good thing. It's a very predictable sign, so you are able to mitigate and take care of patients better.
So wanted to ask, the median is 7 days, but what percentage of patients actually had onset? I think you do have a wide range. Like, what percentage of patients actually onset is after 72 hours? And I think the reason is because that, in the context of potential outpatient setting, that could be very important.
Okay. Let me take that, and then I'll give it to you. So, we do not see CRS right after the infusion or within the first 72 hours. Yes, in the early days, somebody had some fever at day 2 and they called it, etc., because they were all heightened to it. But we looked at it. We said that if somebody is having a fever after the infusion, you better treat them with the antibiotic. This is not due to CAR T cell expansion from cell to cell. That is not the cell to cell signature at all. So it typically median is around day 7, so maybe 5 days. So you are able to treat cell to cell patient in the outpatient setting initially, and start proactively admit them after day +5 if you want, or if you have a facility for 24/7, like in our center, infusion center, they can come to, and then they could be immediately triaged and admitted. They could be followed. So the time is 7, so it gives you the liberty to monitor the patient outpatient. There is no infusion-related fever or within 24 to 48, which is completely different from ide-cel product. Yeah. You have one more to the company.
Yeah,
you still hold on to
Sorry
it.
about that.
Actually, I want to add a little bit more detail about this patient. So this patient had 7 prior lines of therapy. History of also ongoing peripheral neuropathy at study entry when he came into CAR T1. And then he did have grade 2 CRS and also grade 3 ICANS after the infusion of cilta-cel. On day 14, as Dr. Jackman pointed out, he started to experience cognitive slowing, gait instability, neuropathy, but these are all grade 1. He only had grade 3 tremor. That's it. We did also do a subsequent lumbar puncture, and we didn't find any CAR-positive T cells in the puncture.
Thank you. Just sorry, one last question. So our channel check suggesting, I think maybe 1 or 2 sites, they're showing 2 to 3 patients per site per month. I'm just wondering, does that apply to all your active sites so far regarding the supply?
I'll ask Steve to comment on that.
Sure. Can you hear me okay? Yeah, I think the question is on slots per site. So no, there's not one size fits all in terms of the number of slots per site. We have an algorithm that was developed by both Janssen and Legend in terms of how we determine the number of slots per site. As you all know, every site's created a little bit different in terms of the patient volume that they treat. So we have to take that into consideration. So no, there isn't a one size fits all, 2 slots per site. It's based upon an algorithm that we created.
Thank you. Justin Zalin, BTIG. Just a question about the dynamic between inpatient versus outpatient administration. Sundar, you mentioned that it's very predictable about seeing the adverse event profile for patients. Was just curious if you could comment further about how you see the dynamic of unfolding between patients being administered inpatient versus outpatient, and how you see that evolving over time. Thank you.
That's an excellent question. As you know, in United States, every center is different. If you go to Seattle, they will do most of it outpatient. Any CAR T, they are set up to do outpatient, right? And then there are other centers, they have to do it as an inpatient. So there will be, first of all, differences how the institutions are set up, and if the patient gets into difficulty, how they will. But my gut feeling is, as more centers get more experience, okay, then they would feel comfortable if the patient has, just like an outpatient transplant, there has to be somebody with the patient staying and close by so that they could be monitored and then they could come. Some of the patient would rather feel, because they are already into CAR T, and this is being given in relapse refractory situation right now. So these are patients who have been heavily pretreated. This is not newly diagnosed or earlier patient, not a consolidation CAR T. So right now commercially approved CAR T is in relapse refractory, and the patients who are coming through our gate or really trying to make sure whether they can get in, squeeze in kind of a deal. So under those patients, there is a heavy tumor burden and symptom burden. So when you have symptom burden, for me, this boardroom discussion is useless. I don't want to keep the patient outpatient because the symptom burden and others, my patient safety, because I want to make sure I don't get a heart attack and end up in ICU. The patient getting sick and going to ICU is one thing, but I don't want. So you got to remember, I have to preserve myself too. So I think the decision making will change as the patient profile changes and the comfort level goes up.
Yeah. Do you mind if I comment a little bit on that topic? So your gut is right in terms of where the market is heading. So we do a lot of research, as you can imagine, around this topic of inpatient, outpatient for a lot of different reasons. And we're seeing clearly on the research that we've been doing is exactly what Dr. J was talking about. You're seeing, as you would expect, a slow adoption in market right now, just making sure that the staff at our sites are getting the muscle memory down and understand how these patients are reacting to ensure that what we're seeing in the study is what's actually being seen in the real world. But we are forecasting a very aggressive uptake in the outpatient setting. There's no question about that for a number of reasons. But yes, it's inevitable. And one of the features of this asset is this delayed CRS. And you've heard that because of that, you do have now the ability to now wait if you need to admit later. You have the ability to do it at a later point in time. So that frees up a lot of resources, et cetera, and to do that. So, yeah.
And also feedback, because hospitals' infrastructures, now you have 5 or 6 CAR T-cell therapy approved, and hospital infrastructure cannot afford to probably in the future hospitalize all of these patients. So product like this, probably will go in outpatient compared to other products when CRS starts day 1 or 2.
Okay. No, we're doing webcast, so just wait for the microphone.
Thanks. I was just wondering if you could just comment. Obviously, there's a pending approval of a bispecific, and CAR Ts are still in short supply. Could you just talk about sequencing of agents, how you're thinking about using bispecifics in the treatment paradigm?
That's an excellent question. And, we had already reported, and actually publications are coming out too. We had shown that use of bispecific failures being treated on CAR T. Was it me?
No.
Okay. So bispecific patients treated on CAR T when they failed bispecific, and likewise patients who have failed CAR T being treated with bispecific antibody. And the good news is they are quite effective in either direction. And, there has been a study conducted, and it is not yet reported, where they specifically accrued on, subset. I showed A and B. There is a C group also where they accrued specifically patients who have been exposed to BCMA-directed therapy, either ADC or bispecific, to be treated with cilta-cel. So you will get more results. So for me, you are absolutely right. The bispecific antibody, once it gets into the market, again, the question would come. It also has CRS, and typically they have been admitted for the first couple of infusions. So their intake would not just immediately go into the community. It will first get into the academic center, so to speak, but eventually the community will reorganize themselves. Large practices can easily reorganize themselves to take care of these particular patients. So the bispecific will go there. But the most important for me is bispecific, at least the ones which have come so far. I do not know exactly the results of this particular bispecific, but in general, they have the response rate in the neighborhood of 70%. The complete response in MRD is lower. And the PFS is about 1 year plus minus, So they would all eventually have to come back. So it gives you an ability to give that, to buy some time, and then later on they can still go into the CAR T cell therapy. So it's not like one or the other and the other one has taken away, because so far bispecific has not really shown that their PFS and the depth of response is as potent as the cilta-cel.
Maybe Dr. Jagannath, for you, as you think ahead, are you guys prepping for CAR T moving to second line and first line? Do you have enough capacity, or is this going to come at the expense of transplants? I know it's a very sensitive topic.
No, it's not a very sensitive topic. When you're taking care of a patient, you are trying to do the very best for the patient. Whatever cures them, fine. I mean, I got to tell you, since we introduced total therapy from Arkansas in 1989, 89-001, that was the protocol we started, Balagi and I was the chief of transplant there. It transformed at least the life of a younger patient, and we have always shown there is a tail. In that younger population, there have been some patients who have been cured, even though we are supposedly not talking about cure in myeloma. There is a cure. So the auto transplant is there. How auto transplant and CAR T cell will play, because you can use auto transplant followed by CAR T so that the side effects of CAR T goes down. You can challenge auto transplant versus CAR T, But then there is also the bispecific. So there will be a number of permutation combination looking at which one gives complete remission, MRD negative, every year for a minimum of 5 years. And then we can say that we can consistently cure myeloma. So I think at this particular time, I don't think it is a matter of one replacing the other. And also, even now, when we are doing CAR T cell therapy in the younger patient, we recommend that the patient, even if they are going to go straight for CAR T, they still get their stem cell harvested and stored. In case there is delayed hematopoietic recovery or something, you can rescue them with an auto transplant. So I think there are many ways to look at it. Those studies are all being done right now. In the earlier cases, randomized trials are up and running in 1 to 3 lines of therapy and even in frontline therapy. So those are all important questions what you're asking.
And some of the studies with transplants have obviously been followed up with by RevDex and maybe we'll see Darzalex maintenance. But with CAR T, I think some of the initial studies are not doing maintenance. So my question is that even a fair comparison?
No, this is important because for the first time, as we follow, I was impressed because when we originally projected, and ide-cel was ahead of cilta-cel, and ide-cel we were reaching a median PFS, even in the highest 480 million, or 450 million cell infusion, there was about 12 months. Okay, so we were projecting at 1 year when cilta-cel, that's when they said at 21 months the PFS will be 21 months or something like that was projected. But then, as I showed you, I was impressed that my patients who are in remission now coming at 4 years in August, and a couple of patients coming now in June and July. One of them is still MRD negative at 3 years. Another patient is going to be also MRD negative at 3 years. So there is a change in the PFS. Before, you can kind of mathematically predict what the PFS would be. So the question is, will there be a plateau? Could it be? If we see something like that in a relapsed refractory situation, I have a feeling in the earlier phase that CAR T cell. So I'm very happy that for various reasons there was no maintenance after the cilta-cel 1 treatment. So at least we can see what the natural history is, because that will give the strength for physician to design a study with or without maintenance. Because otherwise the knee jerk is myeloma continuous therapy. We got to do something. Oh, the other arm is going to get that. We have to balance this arm too. We should give something more. So that is the general trend, because everybody wants to hedge a bet. But I'm happy in cilta-cel, in a CAR-T 1, we did not do the maintenance, so we can see the natural history, so that it'll, 1 more year, then it'll change the thinking of the physician. If we see there is fewer and fewer relapses and the actual curve changes, the rate, then that'll give the confidence for people to design studies where they don't have to have a maintenance after cilta-cel.
This is David Dye from SNBC. I'm just curious, Dr. Usmani, about your view of those two patients who did not respond to Carvykti. Is there any commonality between these two patients?
You mean in the CARTITUDE-2 trial?
Sorry, CARTITUDE-1. So the response rate is 98%, right?
Yeah.
But 2 out of 97 patients did not respond. So the subgroup analysis was great. We saw however you slice or dice the data, it looks there's response. But what about those two patients that did not respond? Any kind of commonality, common characteristics behind those two patients?
I cannot tell that I can recall anything special on those particular two patients. Do you know anything more detail on the two patients?
I mean, it were not like particularly different patients. So it was.
You should ask any other multiple myeloma drug developers.
Got it. Helpful. Another question just around the CARTITUDE-4 trial. Maybe just can you provide some additional color on the potential update from CARTITUDE-4, and also any thoughts around the powering assumptions for CARTITUDE-4?
We stand by the statement we made in the beginning of the year. That is, we're still expecting potentially the top-line data from CARTITUDE-4 by end of 2022. There's no new update or anything like that. Unfortunately, we're not at liberty to disclose about the powering assumptions and everything.
I can add also, we have disclosed, right? So we recruited these patients, like all patients in a trial. It was last summer, I think August or so. And then with a control arm, which what it is in the protocol. So literature gives 10 months PFS for control arm. So that's the standard of care, 10, 11 months.
Minutes, so we can take maybe one or two more questions from the audience. Okay.
Jeff?
Jeff Meacham, B of A. Dr. Jagannath, I wanted to get your perspective. MRD negativity has been, a evolution, and I want, just in terms of a regulatory endpoint, I want to get kind of your perspective, where you think we are from maybe an FDA perspective about using that, as you move up, upstream in the paradigm as an endpoint, and maybe what it would take to, to kind of utilize that as opposed to a harder endpoint. And that's obviously a gating factor, and it helps speed up development for Legend and for everyone, really.
Yeah, no, that's an excellent question. I do know that the myeloma community, physicians and pharma together went in front of the FDA. First statement was MRD negativity as a goal, as a surrogate marker for PFS, which obviously because the patients were relapsing, and the MRD field itself was by multiparametric flow cytometry. Then you went into the next-gen sequencing. It was tenfold better than multiparametric flow cytometry. They developed the Euroflow, and there was a lot of push from San Miguel and others who had developed from Spain. But all said and done, the Euroflow or multiparametric flow cytometry could not hold the strength to the next-gen sequencing. The next-gen sequencing had the difficulty that if you didn't have the tumor, and even if you had the tumor, in about 10% of the cases, it fails. So these were the teething trouble. But the FDA did approve the next-gen sequencing in this country, and it is Adaptive Technologies has it, so you can get the MRD negativity now done regularly. The next thing is, then of course IMWG, they all get together. Hey, what is good? So the Kumbaya statement was, if they are MRD negative for 1 year, sustain MRD negativity, that would be the great. So then they went in front of FDA, but 1 year MRD negativity sustained did not prove because they were still relapsing. So the FDA did not approve it, okay? So at my center, I'm doing MRD negativity every year for 5 years and continuing maintenance therapy for 5 years and then stopping it at about 5 years, and now I'm looking at those data. And even that, when I started out, because the next-gen sequencing was not available, it was Mayo MRD. Now I'm doing next-gen sequencing MRD when they are coming back. I find out some whom I thought was MRD negative every year, by the time I do the 4th year by next-gen sequencing, I find out they have 11 tumor cells still there. So they are not truly MRD negative. So I have to follow. Then the flip side of it, the physicians who don't do systematic evaluation like I do, they say, what does the MRD mean? Because I have patients who have small amount of M spike. They behave like M because, and I have patients who are 10 year out and things like that. That is another impediment to the field for me, because you can't move the field forward if you don't come to a definition what is considered cure. You have to have a definition for cure. If the expert is going to say, oh, well, MGUS-like phenomenon and those patients don't need to be treated, at the end of 3 years or 5 years, you are still left with an M spike of 0.5. You're going to look in the patient in the eye and tell him, you are cured? You cannot tell that because the patient knows the M spike is still there. You can say MGUS-like, but that would take 10 years to tell retrospectively you behaved MGUS-like. So that is another, crutch the physicians in the myeloma, have to give up and come together and agree MRD negativity and sustain MRD negativity. And that's why, for me, there are clinical trials which have been done. IFM Dana-Farber trial, where they are, read out, it's in the ASCO and it'll be in the New England Journal of Medicine. Their follow-up, they're going to be doing MRD negativity, sustained MRD negativity. So we will slowly hear from cilta-cel and some clinical trial. Now, if everybody, ongoing clinical trials are done with MRD, my gut feeling is if you are MRD negative at 5 years, you're probably cured. And of course, if you follow those patients for 10 years to 2030, you'll find that 10% of patients might have relapsed. But for me, that was a good end mark, and so that the field can move forward for the cure. But these are the two impediments. Number one, sustained MRD, the IMWG criteria 1 year apart is useless. And the second one is you have to give up the clutch going to different. Oh, I have patients who are MGUS-like. So till the physicians give up that clutch and they come up with the definition, thereby they work towards it, then we'll make a progress.
You can do one last question.
Tony Ran. I would classify myself as an independent analyst. Today is my last day at JP Morgan, actually. Just a quick one on ICANS. I noticed that there are a couple of delayed cases of ICANS. Do you guys have any sense to predict which patients, what kind of patient characteristics will make them more likely to have to develop ICANS? Neurotoxicity?
So ICANS clinically is, happens, first comes the CRS. Most patients have CRS, but they could be dissociated. A lot of literature on ICANS is also on CD19 CAR T cells, because they have much higher incidence of grade 3-4 ICANS compared to myeloma. So a lot of the experience and literature comes from them, but most of the things we find as ICANS is usually seen, it could be delayed, but usually within the first month or so. Whereas in cilta-cel, what we are saying is delayed neurotoxicity and separating them out. These are patients who are developing seventh nerve palsy, peripheral nerve, and that's where this whole motor neuron disorder, tremors, and other Parkinsonism-like things. So I necessarily don't classify them, the delayed neurotoxicity, as an ICANS. So I want to say there is delayed neurotoxicity that can happen even 3 months down the line. In our case, in our patient, it was day plus 100. He was diagnosed with Parkinson-like, and there have been another center even later than that. And you heard today we reported a case 3 years down the line, Parkinsonism. So that is different. But ICANS is more due to CAR T cell and its related toxicity. So there is a relationship with the cytokines and other